Hello friends, welcome back to my channel Medicine of 101. Today we'll talk about physiology of thyroid gland part 3. So in the first two parts video I have already described how the thyroid hormone is getting synthesized within the thyroid gland and how it's getting circulated from here within the circulations and how they are getting binded with your circulations with the different kinds of the proteins, the binding capacity and the half-life of the T3, T4, all these things I have already mentioned in the previous two videos. Now in this video basically we will talk about the regulations where from the regulations, that means in terms of the hypothalamus, pituitary and the thyroid gland in terms of the regulations of secretions of what? Thyroid hormone, that means the T3 and T4. Okay. Now see, whenever we are talking in these things, basically we know that for example is the hypothalamus, and just under the hypothalamus what is there? There is the pituitary gland, right? So if we see the where is exactly the hypothalamus, if we see where exactly the hypothalamus, for that thing if we just draw a little bit of the central nervous system. Okay, let's see. For example, within the forebrain we know that there is two parts, orthoncephalon and what? Telencephalon. That means we can say over here the diencephalon and what? Telencephalon. So see over here within the telencephalon we will get structures from here, for example, and these structures. Okay, this is the hypothalamus and this is the pituitary gland. Okay. Now, where within the forebrain, I have already said within the telencephalon part. Okay, sorry, within the diencephalon part, we will get this type of the structures. Within the diencephalon part, not telencephalon. Within the encephalon, we will get this type of the structures. What? Hypothalamus, and under the hypothalamus, we will get the pituitary gland. And the same over here. Now, within the hypothalamus, we know this is the mammillary body. This is the mammillary body we will get over here. And these portions, that is known as what? Median eminence. And the portion that is connecting the pituitary gland along with the hypothalamus, that is known as what? Infundibulum. This is the infundibulum over here, this portion. And this is the mammillary body. And these portions outside, outgoing, this is known as what? Mammillary body. Now we'll see what type of thing is happening over here that is regulating the secretions of the T3 and T4. Now, as we know that within the hypothalamus there is the presence of some types of the neurons, some special neurons where within the paraventricular space, within the paraventricular area. So if you write it over here, within the hypothalamus, what? Within the hypothalamus there is presence of what? We get some neurons where the location within the paraventricular area, right? Paraventricular area. Now we will see over here these neurons, the special neurons within the paraventricular area, they can release sometimes of tripeptide. What they will release over here? They can release what? Tripeptide. What is the tripeptide over here? There is a TRH. Okay. Now see, we are getting over here that there is a connections. There is a connections by the circulations in between the what? In between the neuronal cells and the anterior pituitary. Now what is the name of the circulations that is connecting the neuronal cells along with the anterior pituitary? That is known as what? Okay, let me know. Hypothalamic hypophyseal portal system. I am repeating once again. This circulatory system that is connecting the the neuronal cells along with the anterior pituitary over here. If we say over here, okay, then this thing is known as hypothalamic hypophyseal portal system. So what is this thing? We'll get it over here. That is known as, okay, I'm writing it over here. It is known as hypothalamic hypophyseal okay, portal system. Okay, that is connecting the what? The neuronal cells along with the anterior pituitary. Now, if we see it over here, whenever the TRH or we can say the tripeptide that will be released by these neuronal cells over here, that is present within the paraventricular area. Now, let's show the TRH over with these things. Now, the TRH is released and through the circulations, we will see the TRH that will reach where within the anterior pituitary, and within the anterior pituitary there is a special type of the cells, thyroid cell, or you can say thyrotrophs. Okay, within the thyrotrophs, for example, is the thyrotrophs or the thyroid cells. Here we will get the presence of the receptors for this TRH. within the triiodothyronine, within the what? Within the thyroid cells that is present within the anterior pituitary, or you can say the thyrotropes. Within the thyrotropes, we will get the receptors, regions of the receptors for what? TRH. Now, for example, if we get the receptors over here, okay, is the serpentine receptors. Okay. Now, at first, whenever the TRH will come over here, we got over here what? The TRH. The TRH over here. And whatever thing will see within the cells, let's see over here. For example, is the thyroid cells. I'm just showing this thyroid cell over here, okay? Now, whatever thing we'll see. See, whenever there is a presence of, I have already said that the serpentine receptors, this receptor for what? TRH. The receptor for TRH. Okay. First TRH that will be released by what? This neuronal cells through the hypothalamic hypophyseal portal system. The TRH that will reach up to what? The anterior pituitary. Within the anterior pituitary, especially where within the thyroid cells, or you can say thyrotrop cells over here. Okay. Now see, whenever this TRH it will come, it will just get attached with its receptor. As we know that what kind of the receptors this is? This is what? GQ. This is a GQ. Now whenever we will get the GQ, or whatever the thing we will get over here, whenever it will be active, it will activate another enzyme. It will activate another enzyme that is known as what? Phospholipase C2. Phospholipase C2. So whenever see this GQ, okay, receptors that will be activated by the TRH, this GQ will activate another enzyme that is known as phospholipase C2. So if we write it over here, PLC2 stands for what? Let's write the whole form. Phospholipase C2. Now whenever the phospholipase C2 that will be activated, now it will break down on component that is present within the membrane. Now the phospholipase C2 that will break down on component that is present where? Within the membrane, within the cell membrane. Within the cell membrane, there is one famous compound, that is known as what? PIP2. As we know, within the cell membrane, there is the presence of phosphatidyl ionocytyl diphosphate. So within the membrane, we will get these things. Now, what this phospholipase C2 it will do? It will come over here, and it will break down this, phosphatidyl ionocytyl diphosphate, and it will convert into two components. What? One will get diacylglycerol. Another compound will get the IP3. Will get diacylglycerol, and another will get IP3. These are the what? Secondary messengers. These are the secondary messengers. Now see, whenever we will get the secondary messengers, the concentration of the secondary messengers that will be increased within the cells. What they will do? See, they can activate another kind of the enzymes over here. So let's talk about those things. See, this diacylglycerol they can activate another enzyme that is known as protein kinase C. What about the IP3? IP3 that will also activate the protein kinase C, but in case of the IP3, try to understand, it will activate first the endoplasmic reticulum. The IP3 that will activate the endoplasmic reticulum. So whenever the endoplasmic reticulum will be activated, it will release more amount of the calcium, more and more calcium, and this calcium that can also activate what? This protein kinase C. Now see, whenever we are getting this protein kinase C that is getting activated, at that time it will give multiple signals through multiple steps. No, multiple signals in multiple ways. Ultimately, we can say it will increase the synthetic activity of the cell. It will increase the synthetic activity of the cells. And ultimately, we will see over here one signal that will come over here, and from here we will get releasing of the TSH. We will get the releasing of the TSH. In the same way, if we just show over here, at first we got what? The GQ, then it stimulates what? The phospholipase C2. Phospholipase C2, phospholipase C2, it broke down what? The PIP2, for example, the PIP2 into two components that is present in the membrane. One is DAG, that means diacylglycerol, another IP3, and this IP3, okay, and the DAG they will activate the protein kinase C, and the IP3 that will activate first the endoplasmic reticulum, and the endoplasmic reticulum they release calcium. This calcium and this DAG they will activate the protein kinase C. So protein kinase C that will activate the different kinds of transmembranous things, or you can say the transcriptional things over here. So one of the signal that will help to release the TSH, that means thyroid stimulating hormone. So from here ultimately we will get the releasing of TSH. So from here ultimately we will get releasing of the TSH. Now as we know within the, see, what do we know within the follicular cells? It is thyroid gland. Within the thyroid gland, within the follicular cells, there is the presence of receptor for what? TSH. There is a receptors for TSH. Now this TSH that will come over here, we can say this TSH that will go over here and they will stimulate these receptors. Okay, now whatever we get in these cases, let's draw these things over here. Okay, I am drawing it over here. I think that will be easier for you. Okay, I am just showing on cells from here. Okay, on follicular cells from here. Now see, if I just show you on follicular cells, now whatever thing we are going to see over here. See, on follicular cell I am showing it over here. Now as I said it over here that there is a presence of what? There is also receptors within the follicular cells, and that is also serpentine receptors. So we'll get also over here what? The serpentine receptors. So within the follicular cell is the follicular cell, right? Within the thyroid gland. Follicular cell. Now whenever we will see that we already got the productions of the TSH. Now the TSH that will come over here and that will activate the what? Its receptors. So whenever the TSH that will get bound along with its receptor, at that time whatever the thing will see, as we know this is the G stimulatory receptor, G stimulatory receptors. Now whenever G stimulate, this receptor that will be activated, this GS, what they will do, they will activate another enzyme. What they will activate? They will activate the adenylyl cyclase. What? Adenylyl cyclase. Is the word adenylyl cyclase. So whenever they are activating the adenylyl cyclase, at that time what this enzyme will do, it will just convert the ATP into cyclic AMP. It will convert what? ATP into cyclic AMP. So whenever we are getting the more amount of the cyclic AMP, so more and more amount of the TSH is coming over here, more activation and more activation of the adenylyl cyclase, so more conversions of the ATP into cyclic AMP. As a result, the concentration of the cyclic AMP within the cell will get more. Okay. Now, whenever we are getting the activations of the cyclic AMP, what it will do? It will activate another enzyme that is known as protein kinase A. Protein kinase A, and this protein kinase A, through multiple signals pathway, it will do many things. Okay. Now see, for example, this protein kinase A by on signal, this protein kinase A in on signals, it is activating the iodide pump. So whenever we will see the, for example, iodide pump in these directions, of course. For example, it is activating the, okay, let me show this color one. It is activating the what? Iodide pump. So whenever the iodide pump that will be activated, so more amount of the iodide that will be taken within the cells. Okay, for example, another signal that is getting activated and that is increasing the activity of the, for example, this is the enzyme thyroid peroxidase. Thyroid peroxidase. So it is what? Increasing the activity of the thyroid peroxidase. In a nutshell, you can say, for example, another signal. Okay, let me see it over here. That is activating the nucleus. Okay, and it is increasing the production of the thyroglobulin protein. Thyroglobulin protein over here. So in a nutshell, we can say this protein kinase A that will increase the synthetic activity of the thyroid follicular cells. So the protein kinase A we are getting over here. This protein kinase A that will activate what? The synthetic activity of synthetic activity of thyroid follicular cell. So whenever this activity will be increased, at that time what did I say you? There will be different kinds of the reactions. What I have already explained, how the T3 and T4 is getting formed. So whenever we will get the more amount of the T3 and T4 is coming, so ultimately through the also we will get what? More amount of the T3 and T4. Okay, let me show over here. That will be taken by what? That will be coming within the circulation. So we will get what? The T3 and T4 within the circulations. After coming within the circulation, whatever the thing we'll see over here, try to understand. So whenever the T3 and T4 we are getting within the circulation, as we know that within the circulation there is a presence of the different types of the binding protein. Okay, thyroid binding globulin protein. So this T3 and T4 they will come over here and they will get bound. We will get over here this T3, T4, for example T3, T4 they will come and they will get bound. T4, T4 I have already explained. These are the thyroid binding globulins, thyroid binding pre-albumin, thyroid binding albumin proteins. Now see, whenever you are talking in this way, that whenever from the thyroid follicular cells or the thyroid gland, if we just say the secretions amount, so more in amount, if we see over here that T4 that will be secreted more. So T4 secretion, for example, will get around 90%, and T3 will get around, for example, 10%. So here try to understand, definitely whenever we are getting the more amount of the T4, okay, within the circulations, at the time what do we know, this T4 is having the more affinity. So T4 having more affinity with thyroid binding globulin protein, so whenever they are having the more affinity, so they will be less functional. Okay, another thing is over here, this T4 is biologically inactive. This T4 that is biologically inactive. So another thing you can get over here, this T4, okay, this T4 that will be converted by what? 5 prime. I have already explained these things. Okay, please watch those videos, then you can watch this one. Then it will be easier for you to understand. 5 prime D monodeiodinase. 5 prime mono deiodinase. This will convert T4 into T3. Now it will be functional. Now how this thing, that means the central regulations, they will regulate the secretions of the TSH from the thyroid gland. Okay, now we have to understand these things. Okay, for that thing I'll just draw more these things over here. Okay. Let's draw it in this way, guys. Anyway, for understanding purpose, I'm drawing these things. Okay. Now try to understand, as I said you, that there is the presence of what? The thyroid cells over here, right? Thyroid cells. And we'll get over here the neurons. We'll get over here the neurons. We'll get over here the neurons over here. Okay. Now try to understand what type of thing we said over here. We got the circulations in terms of the what? Hypothalamic, hypophyseal, portal circulation. Okay. This is the circulation, right? Now see, whenever we are seeing over here that, as I said, the more amount of the T4 that will be released. So whenever we are getting more amount of the T4 is released over here, so this T4 is having the more capacity to regulate. T4 is having the more capacity to regulation. So if we say the major regulator hormone in terms of the T3 and T4, that will be T4, because T4 is getting secreted more in amount. So the T4, that is the main regulatory hormone, regulatory hormone over here. Okay, now how the T4 that will regulate? Basically, I have already explained you that T4 is inactive form. T4 is the inactive form of the what? Thyroid hormone. Now, always you will see from the circulation some amount of the T4 that will be taken by the what? Thyroid cells that is present within the anterior pituitary. So always some amount of the T4 that will be taken. Within the thyroid cells that is present within the anterior pituitary. So see, if we get a T4 over here, after going within the thyroid cells that is present within the anterior pituitary, this T4 that will be converted into T3, and this T3, okay, let's write it over here, this T3 that can regulate the TRH receptors. This T3 can regulate the TRH receptors. Now how it will regulate? Try to understand. Try to understand. Okay. If we see within the circulation, if we see within the circulations, the amount of the T4 that is getting increased, So whenever the T4 that will be increased, definitely the more amount of the T4 that will be converted into more amount of the T3. So whenever the T3 that will be more, at the time it will inhibit the TRH receptor. So try to understand what I am saying. Whenever the T4 within the circulations that will be elevated, at that time more amount of the T4 that will reach within the anterior pituitary, within the thyroid cell. And after going there, it will be converted into T4, from T4 to T3. So whenever we get more amount of the T3, as I said, this T3 that can regulate the TRH receptor that is present within the thyroid cell over here. Now see, if we get the more amount of the T3, it will inhibit the what? TRH receptors. It will inhibit the TRH or receptor. Inhibit the what? TRH receptors by what? Negative feedback. Negative feedback. Okay. Now if we see rather another thing in the circulations, the T4 level that is getting decreased. So whenever the T4 is decreased, definitely the less amount of the T4 that will be reached where? Within the anterior pituitary, where? Within the thyroid. Try to understand. So less amount of the T3 will be converted. That means we'll get the less amount of the conversions of the T4 into T3. So whenever the T3 that will be less, at that time this T3 that can activate the TRH. See what I am saying you. Whenever T4 is less, at that time less amount of the T4 that will be converted into less amount of the T3 where? Within the anterior pituitary. So whenever the less amount of the T3 we are getting over here, this less amount of the T3 that can activate the TRH receptors. So this will activate the what? TRH or receptors. So whenever it will activate the TRH receptors, at that time more amount of the TRH will come over here, and as a result we will get more amount of the secretion of what? TSH, and the more TSH that will come over within this portions, within the thyroid follicular cells, and as a result we will get the productions of the more amount of the T3 and T4 here. Okay. Now we'll talk about two more important things, that short-term reactions for TSH and long-term reactions for the TSH. Okay. So let's talk about those things. Now guys, whenever I am talking about the short-term reactions and the long-term, what do you mean by this thing? As we know that half-life for the TSH, try to understand. If I write it over here, the half-life of TSH is only 1 hour. That means what? 60 minutes. So for example, is the thyroid follicular cells. Okay, and is the receptors for TSH. Is the receptor for receptors. Okay, I showed you initially. Okay. Now as the receptors for TRH. TSH. So whenever we will see the TSH, because it is having the half-life very less, at that time if we see secretions of the TSH more in amount, they will come over here and they will work very rapidly, and they will give the many signals, as I said over here, what are the things. Ultimately we will get the productions of the T3 and T4 more. So if we see in terms of the short-term reactions over here, at that time, okay, the T3 and T4, TSH will come over here, it will activate its receptor, and we will get the productions of the T3 and T4, because its half-life is around how much? 60 minutes only. But the problem is over here, when we will get the for longer durations, if we get the active TSH, whatever the problem we will see. So now when we will see the longer activations, or the durations of actions of the TSH will be increased at that time, whatever the manifestations we will see over here. Let's talk about that thing. Now see, for whatever the regions, if we see the activity of the TSH, let's draw it over here. Okay. Now see, is the receptors again. Now for whatever the regions, if we get the activity of the TSH on the TSH receptor that is getting increased too much, this activity is getting increased too much. Whatever the thing will see, try to understand from here. Okay. See, these stages, for example, if you talk in terms of the Graves' disease, where we'll see the autoantibodies, where we will get the autoantibodies against these receptors, against the TSH receptors. So instead of that, if we get, for example, we are getting the autoantibodies. We are getting the autoantibodies against these TSH receptors. So whenever we get the autoantibodies, they will give the multiple signals, and they will just give the signals for longer durations over here, right? So we'll get the signals, or you can say the actions or durations of action that will be increased. So whenever we get the durations of the actions, that will be increased. So TSH, that will give the signals over here. As a result, we will get over here the T3 and T4, well, that will be elevated productions over here. And another thing, whatever the thing we'll get over here, whenever I was saying that duration is getting increased, whatever the function will get over here, this TSH receptors, whenever it will be activated, it will give many signals. One signal that will increase the productions of the T3 and T4. Another signal that will increase the size of the follicular cell. Another signal it will go, it will just increase the proliferation. If we see the number of cell, that will also increase. Another signal, not only these things, it will also increase the number of follicles. Whatever the things we'll see, let's see in a diagram. Okay. Let's try to see in a diagram, then I think it will be easier for you guys. Normally, what do you know? Is the follicular thyroid cells, right? Follicular cells over here. And here we will get the cuboidal epithelial cell, right? Cuboidal cell. And here we will get the follicle. In the middle, we will get the colloid, okay? So whenever we will get the actions for the TSH for longer durations, whatever the thing will get over here. See, the number of the cells that will be increased, the size of the cells that will be increased. So whatever the thing will see, if we just show it over here, ultimately we will get, okay, let's use this curve, okay, no problem, I can use this one. We will see in these cases, the shape of the thyroid gland that will be changed, the shape of the follicle that will be also changed, and in these cases we will get the cuboidal cells, they will be converted into columnar cells. The cuboidal cells, they will be converted into columnar cells. Okay, now see in these cases what are the things we got over here. See, in these cases we got the cuboidal cells that is getting converted into what? Columnar cells, right? Columnar cells. And see, whenever we are seeing the crowdance of the cellular activity, we can say the crowdance of the cellular numbers within the follicles. At that time, see the follicles that will be compressed. So whenever the follicles, we can say the colloid that will be compressed. As a result, the size of the follicle that will be narrow. So we'll get the narrow size of the follicle over here. Now, another thing we can get, as I said, the number of the follicles can be increased also. So whenever we are talking in this way, that number of the follicles that will be increased. So whatever the thing will get over here. See. Number of the follicles that will be increased means we will get within the follicular cells. For example, let's draw it over here. Now, in this case, we can get the number of the follicles. See, one follicle here we are getting two follicles, and these cells will be also just like this one, the columnar cells. Okay. We can get the columnar cells. So in these cases, we will get the number of the follicles. That will be increased. So whenever the number of the follicles that will be increased, definitely